Hi, my name is Taylor Garand, and today what we're going to be doing is we are going to be using a chemical and a physical method to control microbial growth in my 21-day sauerkraut brine. So today what we're going to be doing is we're going to heat boil for our physical method, and we are going to use disinfectant for our chemical method. As you can see, I have my safety goggles on, my gloves, my long sleeve and warm pants. Using proper PPE while doing experiments is very important. Before starting my experiment, I made sure to clean my surfaces with the 10-to-1 bleach solution. And what we're seeing here is I had already labeled my agar plates for my chemical brine and my physical brine. And here we have our chemical and our physical brine samples before using our chemicals and our heat. In my disinfected pot, I have here my brine. What we are doing today is we are using our untreated control sample and we're going to compare it against two different cleaning methods to see how well the bacteria stops multiplying. Here we have divided our remaining brine from our 21-day brine sample equally to apply our two treatments. So the first technique is the physical method, where we use extreme heat. What I'm going to do is transfer half of the brine into my heat-safe pan here, and I'm going to bring it to a rolling boil at 100 degrees. Now our brine is getting to a rolling boil here in just a second, and what we're going to do is we're going to let this boil for a whole minute. Once the minute is up, we will take this off of the heat and let it cool completely. This is a physical control because the thermal energy physically forces the bacterial proteins to denature and unfold, destroying the cell structures. While I let my other brine cool, this is the chemical method. So what we are going to do is we have our half of the brine, and we're going to add household bleach to reach a 10% final concentration. What I'm going to do is I'm taking this pipette and I'm going to be adding in all of my bleach. Okay, now that we added our bleach, we are going to swirl it gently and then let it sit undisturbed for 10 to 15 minutes for contact time. This is a chemical control because the active ingredients chemically oxidize and break down the essential components of the bacteria cell walls and membranes. We expect this treatment to drastically inhibit or even entirely eliminate the microbes, resulting in little to no growth on our plates. Here you can see I have my sterile area, and what we're going to do is we are now going to get our samples, and we're going to start out with our physical heated brine. So what we're going to do is the EMB plates and the TSA plates, which select for gram-negative bacteria. Using our strict aseptic technique, I'm going to flame my inoculating loop to sterilize it first. Now I'm going to let that sit for just a few seconds here, about 10 seconds to cool down. I'm going to get my sample of my brine, and I'm just going to open this up just enough to be able to put my sample on them. I'm going to be very gentle so I do not damage my plate. Now, what I'm going to do is I'm going to sterilize again. Now now that this is cooled off, I'm going to grab my sample for this plate. Very lightly put my sample on. You're only going to open this up enough just to be able to get to it. Not let any other bacteria get onto your plates. So now what I'm going to do is I'm going to use the flame to clean off my inoculating loop. I'm just going to let it cool down, and then we will do our chemical shrink plates. So now what we have here is our chemical brine, which has sat for 10 to 15 minutes. And I'm going to put some of my brine with my clean inoculating loop onto my plates. So I had already cleaned off my loop. And I'm going to open this just enough to streak my plate here. Very gently so I do not damage my plate. Now what I'm doing is I'm taking my inoculating loop and I'm sterilizing it. And now that my loop is cleaned off, I'm going to take more of my brine. And I'm going to put it here on my other shrink plate. And now I'm going to sterilize my loop before I set it down. Now that we performed our streaks on our EMB and TSA plates, flaming the loop between every single one to clean it, I repeated this exact flame and streaking process for both my physical and my chemical brine. Now that we are all finished, I had taped the sides of my plates, that way nothing gets in or nothing gets out of them. We are going to let them sit for 24 to 48 hours, and as they incubate, we'll see what we get. We are at the 24-hour mark for my 21-day brine, along with my chemical and physical agar plates here. I'm going to be completely honest, I do not see anything even on the 21-day as of 24 hours. And there is also nothing on my physical or my chemical agar plates. So that is the end of my experiment. Thank you for watching.